So far we have talked about various ways of collecting and processing data, which we acquire in quantitative experiments. And then we also spent some time talking about R, either kind of the beginner level or at the more advanced level. And today and tomorrow, we will focus more on the next steps of data analysis. So what happens after we process the data using tools such as Skyline or FragPipe. And now we are interested in the statistical analysis of these experiments, where we would like to detect changes between various conditions, find which proteins are differentially abundant or regulated, and so on. And so the plan for today and for tomorrow is to focus on MSstats and its interface with the existing data processing tools and on statistical analysis. Today, we will focus on label-free experiments, in particular DIA, and then tomorrow we will focus on label-based experiments, in particular with TMT. Before we start, let me just remind you of the logistics in case you are joining us for the first time. So at the bottom of your screen, you have a chat button. Let us use it just for things such as, hi, how are you? Or I can hear you. And otherwise, if you have questions related to the technical parts of the presentation, please use the Q&A button. The Q&A allows us to manage the questions more effectively, and also it allows us to save the questions and make them available after the session. Several people commented that the Q&A are accessible, but you cannot download them, so I need to change the permission on the files. I didn't realize, so as soon as we're done with the presentation, I will change the permission. Well, otherwise, let us go ahead and get started. So today I will be leading the first part of the presentation. My name is Olga Vitek. I am a faculty in the Curie College of Computer Sciences at Northeastern University in Boston. I have a PhD in statistics from Purdue University, and I did my postdoc in the Institute for Systems Biology in the Abersold lab back then. After that, I went back to Purdue as a professor in statistics, and since the last 8 years, I am now in the College of Computer Sciences at Northeastern. And also joining us today, Mina Choi. Mina is a senior research scientist at Genentech. Mina holds a PhD in statistics from Purdue. And for some time, she also was a postdoc and then a research scientist at Northeastern. Most importantly, Mina used to be the original lead developer of MSstats, and a lot of work that we will talk about today is actually her work. So we're very fortunate to have Mina today as one of the instructors. And let me also introduce Devon Koller. Devon is a PhD student in computer science at Northeastern. Devon is currently one of the lead developers of MSstats. In particular, he develops Shiny graphical user interface so that we can use this type of interface for interacting with MSstats. And we're very happy to have Devon as TA today. Tomorrow, Devon will actually lead part of the discussion presenting the broader MSstats ecosystem. And let me also welcome back Brendan, who was leading the Skyline discussion for today's last week. And so he will also make a bridge between the discussions of last week to the analysis in MSstats in the second part of the presentation today. Okay, thank you so much. So now we will go ahead and get started with the technical material. Let me just, here we go. So I will jump in right away and let's talk about MSstats, what it is. So MSstats is used to be a package, and now it's a family of packages which are based in R and distributed via bioconductor. And fundamentally, this is a family of packages which focuses on a series of tools for quantitative proteomics. at the very kind of core of the question that MSstats is answering is which proteins change in abundance. What is important is that it focuses on various levels of complexity, in particular complex designs such as factorial experiments, paired design, time course, so designs which are more repeated measurements, so anything which is more complex than comparing two conditions. However, you can of course compare two conditions as well with MSstats. A byproduct of this type of answering this type of questions is also quantifying the abundance of a protein in each subject on a relative scale that can be compared between runs. So this type of summaries are useful if you want to do, for example, clustering or use protein level quantities as input to some machine learning tools or biomarker discovery tools. Technically speaking, in terms of the experiments, MSstats can handle a variety of types of acquisition strategies, in particular data-dependent acquisition, either label-free or labeled with TMT or SILAC or something else, data-independent acquisition, also PRM and SRM. So this is the type of experiments that Brandon would call chromatography-based quantification. And of course, also TMT, which is based on reporter ion quantification. More recently, we went beyond just studying abundances of proteins. So there is also functionalities for post-translational modification. So for example, detecting changes in PTM and distinguishing them from changes in overall protein abundance. And then also structural proteomics for limited proteolysis. So there are quite a few functionalities. In terms of the input to MSstats, the input will be the output of data processing tools, such as we discussed last week, so Skyline and FragPipe, but also MSstats provides converters to a much broader variety of tools, in particular MaxQuant, OpenMS, and open source, and also commercial tools such as Spectronaut, Proteome Discover, etc. These converters, they are actually very important. They do multiple things. They not only, take data in one format and transfer it in another format, but also they do various types of quality control, such as checking for the correctness of the input, checking for duplicate features, checking for decoy peptide IDs if you happen to use target decoy for quantification, and so on. We will see some of this during the hands-on session. So these converters are really useful because it means that besides just being convenient, it also means that you can potentially have a same standardized type of statistical analysis regardless of the tool you used to process the data upstream. And this is very important because this allows us to understand how processing strategies impact our statistical conclusions if we can analyze later on all the resulting data sets in a consistent manner. Of course, the core of MSstats is actual statistical modeling and statistical analysis. So we will, and I will talk about this today, we have models which are very flexible, account for complex designs, missing values, outliers, and so on. They're also numerically stable. We pay a lot of attention to making sure that parameter estimation is stable and scalable. A good thing is that a lot of this work is done behind the scenes so that the users do not have to be experts in statistics to use that. And MSstats recognizes just from the input formats, the type of experiments and the type of data, and then fits the right models. And also beyond just quantifying proteins on a relative scale, we have a lot more additional functionalities, in particular methods for system suitability, quality control, assay characterization in terms of limits of detection, limits of quantification. And something which we do already and we're interested in doing more is using our data sets that we analyze as a pilot study for something else. So for the next experiment, we can learn the extent of variability from the current data set and try to understand what would be the minimal sample size and also what would be the optimal allocation of resources for a follow-up experiment which uses the same measurement workflow and the same type of biological material, but maybe wanting to gain more sensitivity or answer slightly different questions. So this is really the scope of MSstats. And before I go any further, I would really like to acknowledge many people who have been contributing to that. So Mina, as I mentioned, was one of the original lead developers, and a lot of work I present today is really her contribution. I would like to highlight Mateusz Staniak, who, now that Mina is at Genentech, Mateusz is the lead developer of MSstats, and also Ting and Devon, who have made a lot of contributions. Ting focused on TMT. Devon has worked on PTM and LEAP and also Shiny. And also Melanie has been a very important collaborator in terms of integrating MSstats with other interfaces such as Galaxy. So this work was supported in large part by Chan Zuckerberg Initiative, which allowed us to restructure the code to make it scalable and make more robust. If you have been using MSstats in the last few years and haven't used it recently, since the last year or so, the entire code base of MSstats has been completely rewritten to make it much more effective. And so we will hear a little bit today and especially tomorrow from Devon, so how this structure actually is organized. So there are converters, base MSstats, MSstats TMT, and so on. And then there are additional interfaces for interactive interfaces. Okay, so let's start with the overview of MSstats. So what I would like to do is to talk about some really basic MSstats features, and then, as much as time allows, talk about good reasons why we would be using MSstats as opposed to, let's say, a t-test or some other methods. So what would be the benefits specifically for the case of DIA for today? So this is a typical data processing workflow in MSstats from a statistician's perspective. So, you know, oftentimes when people focus on proteomics and data acquisition, there will be, you know, a lot of steps of data process, of sample processing, sample handling, data acquisition, and then there's like a little box, data analysis. So statisticians think about this in the opposite way. There is quite a bit that has to happen before we collect the data, and as much as has to happen after we collect the data. And of course, data collection is in the middle. So I will start from this part, and then if time allows, I will talk a little bit about the method validation and system suitability as well. But first thing, experimental design. This is really very important, because if the experiment is designed incorrectly, then no amount of statistical sophistication can really help us compensate for the flaws of design. And so from our perspective, what is really important is understand what are the goals of the study. In particular, do we just want to do exploratory analysis, some kind of unsupervised analysis? Do we want to do biomarker discovery, which would be more supervised classification? Or do we want to do population level differential analysis? If we had all day, I would tell you more the differences between these methods, but the long story short is that these different goals require different statistical and machine learning techniques. And so MSstats focuses specifically on differential analysis. So if we find proteins which change in abundance between conditions, these proteins are not necessarily biomarkers, for example, and that's really important to remember. And so, and the reason for that is the differential analysis compares population level means, meaning the overall mean of a protein in the untreated population versus the overall mean of protein abundance in the treated population. Whereas the biomarkers focus on the status of an individual subject, and they want to predict whether the subject has the disease or not. So this requires different statistical and machine learning technology. So we focus here on differential analysis. A second really important part here is the biological aspect. So what are the conditions that we're interested in? What are the replicates? The replicates are important to handle uncertainty and variation. And we have two layers of uncertainty and two layers of variation. One is biological variation, person to person. We all have different protein abundances between us. We also have different protein abundances from one day to the next, or from one time point to the next. We also have noisy measurement technology, which introduces additional noise and variation. And so because of that, we need to characterize whether we have replicates on the same subject across multiple time points, multiple biological subjects, or multiple technical replicate runs on the same biological sample. These aspects will require different statistical analysis. And of course, technological aspects such as sample preparation, label-free, label-based, multiplexed or not, the type of data acquisition, DDA versus DIA, the randomization strategy, these are all very important aspects of the experimental design. So here's one example, kind of older experiment that we had. This was our collaborator comparing two breast cancer cell lines. Now these cancer cell lines were exposed to different level of oxygen, like regular oxygen versus low oxygen. This oxygen exposure was done for different durations. For each duration, there were multiple cell cultures, and for each culture, there were multiple technical replicate runs. Statistical model has to distinguish these different levels of variability, build them incorrectly in the model, and make sure that our conclusions reflect the right level of uncertainty. So this is what MSstats is doing behind the scenes. Here's another example, which is a time course experiment. This was a study on childhood osteosarcoma. So this is childhood bone cancer. It is an example of a hybrid design. So here there were some healthy individuals at one time point, and then there were some diseased individuals which had many time points before the treatment, during chemotherapy, before surgery, after surgery. And you could ask questions. Which proteins change in abundance between healthy and disease at diagnosis? Which proteins kind of become more similar to healthy levels after the treatment? Which proteins change primarily after surgery? So this is the type of experimental design, the type of questions that MSstats can answer. And again, it requires a fairly specialized statistical models to reflect different sources of variation. Well, so once the experiment is designed and data are collected, then a really important part is data processing. As I mentioned, we are equal opportunity. We're trying to be as inclusive as possible with various types of data processing tools. The converters pay attention to this type of quality control. However, a lot of these decisions are somewhat heuristic and somewhat subjective. These converters, they have parameters and so on that you can filter things out or keep things out or keep things in. And so it is really important for us to understand what are the implications of these steps of data processing on the final conclusions of statistical analysis. And we do know, I think it's pretty clear that these steps will have implications for the final conclusions. And our goals as statisticians and as developers of MSstats is to make sure that these biological conclusions are as little dependent on these heuristics upstream as possible. And so they reflect the underlying biology as much as possible, and they reflect the specialized heuristic choices we made upstream as little as possible. And this is really one of the main focuses of MSstats. So here's what the input to MSstats looks like. You will see this quite a bit in the hands-on session. Essentially, we have one row per peak intensity or fragment intensity in DIA. And so here is the intensity of the feature. All these features come from run one. They all come from the replicate A in condition one. So this is if it's a labeled experiment that describes what kind of label there is, what is the fragment and product, and what is the protein name and peptide sequence for this particular feature. You will see this quite a lot. And so the goal of MSstats is to take all this evidence which pertains to one particular protein and summarize it in a way which is robust to all of these artifacts, but also reflects the experimental design appropriately. So let me now walk you through a little bit the data processing, and then we will focus on statistical analysis. So as part of data processing and quality control, we would like to do some visualizations, and tools such as Skyline and FragPipe do this already, but we also do it on our end as an extra check. And so this is one example of a QC plot that MSstats would generate. So here the x-axis is mass spectrometry run. This particular experiment is a time course with 10 time points, and the vertical lines separate time points. The actual order of acquisition was randomized, but now for the purpose of presentation, we reorganize them by condition. Each time point has 3 replicate mass spectrometry runs. And so the y-axis summarizes in the box plots all the log intensities of every feature, every protein, every peptide in that run. And so what we see is that as time progresses, the intensity goes up. And so of all the proteins. And so the question is this biology or is this technology? And the answer is it can be either, right? It depends on the experiment. So the plot by itself only tells us that the intensity goes up, but we don't necessarily know why. Now, this particular experiment happens to have labeled reference standards. So now I can tell you more a little bit. It's a targeted experiment which targeted only a limited number of proteins, which are expected to be affected by a particular stress. And for each endogenous protein of interest, which was targeted, there was a heavy labeled reference counterpart, which was spiked in the biological sample. The reference counterpart is not supposed to have any biology. It was spiked in as constant concentration as possible. And so now, if we do have this type of labeled references, MSstats makes a plot like that, where these are the endogenous intensities here, but these are the labeled reference standards. And we see that the labeled reference standards, they have some variability. This is the technical variation due to the data acquisition, but not to the same extent as the endogenous intensities. So what MSstats would do, it would equalize, in the case of labeled references, the entire distributions of reference standards, and for each run, learn the extent of shift and apply this shift to the endogenous intensities. And so now we see that indeed this shift in intensity is likely due to the underlying biology. And this makes sense because it's a targeted experiment and most proteins will be expected to be changing. So using this type of labeled references is very useful. The problem with that is that sometimes we cannot necessarily add these labeled references for every step. In particular here, these references only reflect the variation which took place during data acquisition. So they do not account for variation due to the digestion or due to sample storage or anything else prior to that. So some of this variability can still be due to artifacts in the samples. Also, we may not necessarily have these standards for various reasons. By the way, just as a parenthesis, this experiment had labeled references for every endogenous protein. It doesn't have to be the case. You just can have like a small number of reference proteins. Increasingly, we see that IRT standards, which are spiked for retention time purposes, could be used as references for normalization, provided that they are spiked in a constant concentration. So there are many other ways to find standards. Ideally, there will be some endogenous proteins which we know are not regulated, and those can be used as standards. Unfortunately, I don't know if such a thing exists, because we never know that something is never changing under any circumstances. So that's very hard to assume. Now, here we really rely on reference standards because the number of proteins is fairly small. I think it's a couple hundred proteins only. But if we're talking about a DIA experiment which quantifies, I don't know, 5,000 proteins, it is more reasonable to assume that the majority of the proteins will not be affected by the stress and only a small proportion of the proteins will be affected. In this case, it would be meaningful to equalize, for example, the medians of the endogenous protein abundances, because we can assume that the median intensity is stable. And if we see some changes, it's an artifact of data acquisition. In this case, MSstats has an option of equalizing the median intensities across all the runs. The choice of normalization, I would say that it's probably the most difficult decision in this type of analysis because it is very hard to be automated. It really depends on the nature of the experiment, on the nature of standards, how many proteins are quantified, how many proteins are expected to be changed by a particular stress. So this is the step which requires most expertise and most care. And I would say that if you see differences in reanalysis of the same data set between publications, very often it's due to this upstream processing and normalization steps. So this is quite important. Okay, and so then after that, we are ready for statistical analysis. And to make long story short for now, the output of the statistical analysis will be fold changes, or actually log fold changes. We do everything on the log scale because it has better statistical properties. So log fold changes and p-values. Log fold changes, this is what we call them as practical significance. It is fine to say I'm not interested in anything that doesn't have at least a two-fold change or whatever cutoff you want to use. However, log-fold change alone is not a good criterion on whether this change that we observe is more systematic than what we expect to see by random chance. We need to compare this fold change to the extent of the underlying biological and technological variation, and p-values allow us to summarize that. So here, p-values are adjusted for multiple testing. I will talk about this in a minute. And traditionally, we take the negative log transform of this adjusted p-values so that high values means more statistical significance. So we are interested in the upper left and upper right corner of this comparison plot. So vertical lines delineate the fold change cutoff. It's fine to apply it as long as we apply the statistical significance cutoff corresponding to the false discovery rate of whatever you chose before analyzing the data. Now, if we have more than one comparison, let's say we have a time course just as before with 10 time points, when we compare time point 2 to time point 1, time point 3 to time point 1, and so on. So we can visualize this in this type of a heat map. The Y or the columns, I guess, in this heat map are comparisons in time. The rows are individual proteins, and the color is the p-value. So it means that blue is significantly down, red is significantly up, black is no change. It can kind of give us the pattern of changes across multiple time points. So this is the type of visualization we do. And of course, the last thing is we would need to talk about experimental design, so that now that we learn how much biological and technological variation is in the data, we can plan the sample size for future experiments or allocation of resources. So now let me tell you a little bit about t-test and p-values. I know that many of you have seen that and are familiar with that, but I also noticed that frequently we use these terms without really kind of being on the same page of what it actually means. And just for the sake of making sure that MSstats is used for the right question, I will go ahead and review very briefly t-test, p-values, and multiple testing, just to make sure we are on the same page. And then I will talk about more specifically about MSstats for DIE. So first, this is not MSstats. This is a very kind of classical standard t-test. So imagine a label-free experiment, which is really, really simple, so that we have for one healthy individual for this protein, we only have one peak. And here is one disease individual, and for this protein, we only also have one peak. So we want to know if this peak is changing in abundance or in intensity between healthy and disease more systematically than by random chance. If we now have multiple replicates, we can visualize this like this. These are healthy subjects, and so this is the log of the abundance of this peak across multiple replicates for the healthy, and this is the log of the abundance for the disease. And so our question is whether yes or no, on average, in the population, the abundance of the healthy is the same as the abundance of the disease. So this is what I would like to emphasize. We are comparing the averages of protein abundances in the entire underlying populations. Now, if the protein is changing on average, it doesn't mean that it has good predictive power for one person. But in terms of screening many proteins for their relevance to a disease, or in terms of just doing basic science and understanding what is affected by the stress, this is totally appropriate. And so that's what we would want to do. Now, the problem about the populations is that whatever the mean in the entire population of millions of subjects who are healthy and millions of subjects who are diseased, we never know what their population mean is. We will never know what it is, but we can make guesses, right? So we can assume something. And so what is easy to assume for us is that on average in the underlying populations, the two protein abundances are the same. There is a convention in statistics that we use Greek letters such as mu for things that we never observe. So in large populations, we never know what the protein abundances are. Let's say mu1 is the population mean for the healthy, mu2 is the population mean for the disease. We don't know what they are, but let's just make a wild guess and say that they are the same. So the difference is 0. And this is what is called the null hypothesis. So we hypothesize that. And then the alternative is that, well, it's something else, right? So there is indeed a regulation. So those two means are not the same. So now we do not know what this population means are. What we do have is several measurements we take on some subjects selected from the healthy population and some measurements selected from the subjects selected from the disease population. And so now we'll try to summarize what we have and what we observe and use this to make conclusions about what we do not observe. So this is what statistical inference actually means, is to take what we observe, summarize it, and make statements about what we cannot observe. So what we observe is a few replicates from each condition. What we do not observe is what happens in the entire population. But let's try to make some statements. So for that, what we do is essentially we build a signal to noise ratio. Our signal is the difference between the group means. So we do not know what happens in the entire population, but we know what the means are in our data. So this will be the mean in the first group versus the mean in the second group. And the noise is how much variation we expect to see for each of those two means. And so now this is a little bit tricky, so I'll just take a minute for that. So this S1 square and S2 square, these are just variances in the populations, right? So sample variances, this is how we calculate them. The deviations from the mean squared, summarized over all the data, right? So this is the sample variance that we see in the first population. This is sample variance that we see in the second population. This is fine, except that this is not quite what we need, because we want to know how confident we are in this mean versus how confident we are in this mean. So it's not the same. So the process for that is like this. So let's say we have a normal population. Let's say we have 3 replicates. We take 3 replicates, we calculate the mean. Then we go back, we take 3 more replicates from this population, calculate the mean. We go back, we take some other 3 replicates, calculate the mean. So these histograms, this will be the histograms of the means of 3 subjects that we take repeatedly from this population. So you see that this histogram is narrower than the original histogram from which we collected the data. Now, if our sample size is larger, let's say here, sample size is 20, we take 20 samples from this population, calculate the mean. We take 20 other subjects from the population, calculate the mean, and then do the histogram of the means. We see that this histogram is even narrower. So the variability or the spread of this histogram of the means is called standard error. So standard error tells us how much uncertainty or how much reproducibility we have in the mean of our data if we were to repeatedly go back and collect the data and repeat the experiments. So this will be the spread of this histogram for the first population. This is the spread of this histogram for the second population. The square root is the overall uncertainty associated with our signal. So one thing, if you are to talk to statisticians for whatever reason, make sure you are clear with the difference between standard deviation and standard error. So standard deviation tells us about how much variation we have in the population. Standard error tells us how confident we are in the mean of the population. And so, by the way, let me also kind of preempt the question. Maybe there is a question like that. So my data kind of look normal here, right? My cartoon. And oftentimes people say, yeah, but mass spec data are not normal. It has outliers, it has some skewness and so on. Is it a problem? Well, turns out that when we're interested in the means of the data and not in the actual populations, it's much less of a problem. So here you see very different distributions, like skewed, uniform, irregular. And let's look at this one, really weird distribution. And we repeat the same exercise, right? We take three replicates, calculate the mean, 3 other replicates, calculate the mean, and so on. Plot the histogram of these means, you see that it's much more bell-shaped than the original distribution. And if we increase the sample size, it's even more bell-shaped. So the means themselves and their uncertainty is much more normally distributed than the original population. This is called central limit theorem. And this is what allows us to oftentimes make the assumptions of normal distribution regarding the means, even if the data themselves are not quite bell-shaped. This said, you see that if we start from the normal distribution, those means are much more reproducible for 20 replicates than if we start with something irregular. So there is an advantage when our starting population is closer to normal. This is why we take a log transformation of the data so that the starting point is closer to normal distribution. Okay, so let me just review, right? So we assume that there is no difference between the two population means, or this is the null hypothesis, or maybe there is a difference between the means. That's the alternative. And so we built a signal to noise from what we observe. So now we need to take what we observe and make some statements about what we cannot see, so about our null hypothesis. How can we actually do that? Well, turns out that if our data are normally distributed, and if in fact the null hypothesis is true, so this assumption actually verifies, then we can show mathematically, without any additional, experimentation, what would be the plausible range of the signal to noise ratio. And this plausible range takes the form of another probability distribution. It's called the student distribution. It's very similar to normal, just a little bit heavier tails. So let me repeat that. So if there is no difference between the population means, then I can tell you what is the range of values that I expect to see for this signal to noise ratio. So this is this particular distribution. And so now what I can do, I can actually overlay what I observe on top of this distribution and say, is it consistent with what I expect to see or not? So if the signal to noise, I put it on top of this distribution and it's somewhere in the middle, you say, yeah, well, that's what I kind of expected to see if there was no difference between protein abundances. So yeah, there is no contradiction with my assumption of no change. On the other hand, if I overlay this on this distribution and it's somewhere very far away from what I expect to see, you say, wait a minute, it doesn't agree. So my values are very different from what I expect. What can be wrong? Well, one thing that can be wrong is that we assume that there is no difference between the two populations, when in reality there is one. So if every other assumption is correct, then it means that I reject the null hypothesis. One additional assumption I forgot to mention is that we assume that each of these data points are independent. So there's no brothers and sisters in our data set, that there is no kind of some other types of similarities which are hidden. So if these assumptions verify, then we can say that we can reject the null hypothesis if the observed signal to noise is not consistent with what we expect. So the last piece is, okay, well, but how large is large? So how far in the tail should I be to claim that there is a evidence against the null hypothesis? And this is where the probabilistic interpretation is helpful, because this is a probability distribution. So the area under this curve corresponds to the probability. So we can specify alpha, which is a false positive rate. It's the probability of false positive. So probability of rejecting the null hypothesis when we shouldn't. So if we specify it, for example, at 5%, then we say if my observed signal to noise is in the tail, then I have 5% of chance of rejecting the null hypothesis when I shouldn't have. And so the value of this statistical analysis and making assumptions is really in this error rate, making decisions and being able to quantify how often you can be wrong in these decisions. So false positive rate is one way to quantify how wrong we can be. And so the related concept is the p-value. Now I actually overlay literally this observed signal to noise ratio, and I also overlay the negative kind of counterpart. And the p-value is the area under the curve to the left and to the right of the observed p-values. And so if the p-value is less than alpha, this is another way to say that my observed signal to noise is too extreme. And so if the p-value is less than alpha, then I reject the null hypothesis. So that's really all there is in terms of like statistics and hypothesis testing. Well, I hope that the co-presenters can answer some of the questions which come up. And if I need to stop and emphasize something, please don't hesitate to interrupt me if there are other open questions. Let me also talk a little bit about multiple testing, because this also comes up, and I want to kind of clarify what the false discovery rate adjustment is about. You probably know, if you have been working with peptide identification and database searches, you probably have heard about the FDR control at the peptide and PSM and peptide and protein identification stage. So this is not what we're talking about here. Our input takes these identifications, these methods take these identifications as input and kind of set it aside. And now we want to focus on the error rates of detecting changes in abundance. Of course, all of those error rates accumulate, but the state of the art currently is to separately control FDR at the identification stage and separately control FDR at the quantification stage. There have been methods attempting to combine that, and I think there is a value in that. The problem is that computationally these problems become very difficult to scale to large data sets and to complex designs. So for the moment, kind of the default strategy in most experiments is to separately control FDR at the identification quantification. So this is what's going on. So remember the signal to noise I just talked about, right? To test the hypothesis. So this was for the case of one protein. Now, what if we have two proteins in the data set? So we will have to repeat this procedure separately for protein 1 and then separately for protein 2. So here I'm trying to visualize this. I just flip this distribution. So this was my reference distribution for protein 1. And this is the reference distribution for protein 2. For protein 1, this is my rejection region. So this is the region when my observed signal to noise is too large and indicates the evidence against the null hypothesis. And so I reject the null hypothesis for protein 1, regardless of what happens for protein 2. So these red areas here together, they're alpha, right? So they have 5% or something like that. So that's fine. But now I do the same thing for protein 2, right? So for protein 2, I'm comparing signal to noise for protein 2 against the same area. And I reject the null hypothesis for protein 2 if, again, I am in this red area, regardless of what happens for the first protein. Now, the problem is that we have both proteins in our experiment, right? So the actual possibility of rejecting the null hypothesis, so claiming that something is changing when we shouldn't, is actually a combination of these two areas. And if we combine these areas, well, together they're greater than alpha. So the probability of making at least one false positive decision regarding changes in abundance is larger than what we would like to claim. So it's larger than alpha. And of course, our experiments do not have two proteins, they have thousands of proteins. So the probability of making at least one false positive decision is essentially 1. So we're guaranteed to, if we try hard enough and test long enough, we always find something just by random chance. So it is very easy to have a lot of false positives just by testing enough proteins. So what this means that we have to be more careful, right? And being more careful here really means more conservative, right? So for example, if we change the decision boundary as shown with this dashed line and only rejected the null hypothesis if we are in this outside region of this dashed line, then we could make sure that the area outside of the dashed line is 5% or whatever we want it to be. Now, the problem is that we see that now we push this decision boundary far back, and it means that now it requires much stronger signal to noise. So much stronger evidence against the null hypothesis to claim differential abundance. And the more proteins we have, the more conservative we have to be. In particular, you probably have heard about Bonferroni approach for multiple testing. So Bonferroni takes this alpha, divides by 2, and then divides by the number of proteins. You have to have a super strong evidence, super large signal to noise to claim differential abundance. Because of course, the best way to not make any false positives is to not find anything. Then we're fine, right? But that's not what we would like to do. So essentially, we have to be very careful about how we control for this multiple testing without being too conservative. And so some 15 or so years ago, there have been a shift in mindset in the statistical literature saying that, well, maybe instead of trying to push the decision boundary too far back, we can change the criteria. So here, the combined red area says we want to make sure that the probability of at least one false positive is very small. When we test 5,000 proteins, maybe that's too much to ask. So maybe we're okay having some false positives as long as we control their proportion in the data set. And so this is how currently people think about this. So this is the truth. This is how many true non-differentially abundant proteins we have. This is how many true differentially abundant proteins we have. This is our decision. We claim no change. Here we claim the change. The problem is that only this number R is actually observed. We do not know what these numbers are. We do not know what these numbers are. However, there are statistical procedures which allows us to control the average proportion of false positives in the list of proteins which we claim are changing. So it allows us to control V, the ratio of V divided by R. Well, we usually find at least something, so it's V over R. The previous approach, Bonferroni and others I mentioned, would really control that V is anything other than 0 with a very small probability. Now we're saying, okay, fine, we will have some false positives, but we want to control the proportion of these false positives in the list that we claim as differentially abundant. And so this is a different criterion, and the method that MSstats is using is called Benjamini and Hogberg. So we can either change the decision boundary as I was showing here, or we can transform the p-values so that we can plot them on the volcano plot. This is quite standard, and many packages rely on that. It's numerically very robust, so that's what MSstats is using. So feel free to ask questions about that. But currently, if you look at the FDR adjusted p-value cutoff, it means that the proportion of the average proportion of false positives in the list of claimed changes is as specified. So if we select the cutoff at 5%, then we expect 5% false positives in our list. Okay, and maybe now this is a good time to also mention again the experimental design. So one thing I would like to emphasize, and this is what MSstats is paying attention to, is how the sample size depends on the number of proteins that we test and on how many proteins are expected to be changing. So let me just, well, first of all, this is the type of figure that MSstats generates. So this is the fold change that we would like to detect. This is number of replicates that we would need to have to be able to detect this fold change, given the variability, biological and technological variability we have seen in the past data sets. So this is what we would expect in the future data set. So if we have only one feature or one protein, so this will be the sample size. However, if we have more proteins, we would need larger sample size, and the sample size will depend on what kind of proteins. So here, the dotted line corresponds to a more targeted experiment where expect half of the proteins to be truly changing. So this would require a smaller sample size. And this top line corresponds to a more discovery protein where we quantify more proteins and we expect only a small number of them to be changing. We need a larger sample size. This may be somewhat counterintuitive, but let's just remember, right, what happened with this multiple testing procedure. We try to protect ourselves against false positives. So in a targeted experiment, there will be more true positives. So there will be less opportunity for false positives. In a discovery experiment, there will be potentially more proteins which are not changing and fewer true positives. If we have fewer true positives, we have more chance for false positives. And so we need to work harder to protect ourselves against that. And work harder in this context means having a larger sample size. So this is another way to think about discovery versus confirmatory targeted experiments. That the more we can exploring and the less we know about our data, the more sample size we need to have to ensure the same control of our error rates. Alternatively, you can have a smaller sample size and have larger error rates. And this is fine too, if this is what is acceptable for our experiments. So the goal for us is not necessarily to request a large sample size, but more to understand the trade-offs between the type of the experiment and the type of false positives and false negatives potentially that we can have. So that's how MSstats thinks about all of these problems. So now we have half an hour left. So now I can actually tell you a little bit about the methods implemented in MSstats, and there is a lot. So just like, last week we had our speakers who had to skip over many details, we will have to do that too. So I will just have a few vignettes which hopefully convince you that it's worthwhile working with specialized methods for our data, as opposed to some generic methods which are available in R or in Bioconductor or in Excel for that matter. Okay, so the first point I would like to make is that the specialized methods in MSstats focus on between tool reproducibility, so that our conclusions express more the biology and less the choice of data processing tool which we selected upstream. So here is some fairly old DIA data sets, but the same point remains currently for more recent data. There are two data sets here. One is the DDA data set by Jürgen Cox from the original LFQ paper. This is the DIA Bruder data set. Brandon talked about this data set last week. Each data set is processed by Skyline, by MaxQuant in this case, Spectronaut in this case, and also by Progenesis in this case. And this is the work by Mina Choi. So what we see here is that these gray histograms, they summarize the log intensity of everything in this data set. So across all runs, across all proteins, across all features, charges, everything. And the x-axis are the same in both cases. So what we see already is just that the location of the histograms changes, right? So different tools report these abundances on different scales, which is not a problem because we do not interpret the abundance by itself. We interpret changes in abundances between conditions. And so if a tool quantifies the peak by truncating the tails or by the peak apex, of course, it will affect the intensity which is reported. And more interestingly, let's take a look at the number of peaks. So 203,000 for Skyline, 224,000 for MaxQuant, 56,000 for Progenesis. Big differences. Here, 1,000 zeros, here is no zeros. Here is 600 zeros. Here is 33 missing values, here is 38,000 missing values. There are big differences between the tools. Now, this is the number of zeros by Skyline, and this is the number of zeros with MaxQuant, for example. So this means that besides just reporting the intensity of a feature and the differences in that, the tools in particular make very different decisions when things are challenging. So when you have peaks that are close to the noise in the data, you can call them zeros, you can call them NAs, you can call them small values, which you can quantify the noise in the expected area of the peak. Sometimes, for example, Skyline distinguishes, so Skyline uses NA when the peak is present but can't be quantified. For example, the peak has a strong overlap with another peak, or it's truncated, the window has cut the peak for like in a big part. Otherwise, it will probably integrate the area in the expected region. MaxQuant seems to be much more generous with NA and has a lot more NA than Skyline has. And so what we realized when we worked with all of these data processing tools is that we should not be interpreting this type of readouts literally, because sometimes the same pattern in the data is represented in very different ways by different tools. And they have their reasons, and sometimes they emphasize certain things. But if we want to make sure that our statistical conclusions are not that dependent on these choices, we need to read between the lines and understand what was the intention by the tool and try to summarize things as statistically appropriate as possible. So what we decided to do back then is to essentially not pay attention to zeros versus NAs versus low values. And we came up with this cutoff, which is dataset specific, where we say below this cutoff, we don't trust the values. So call them small values, zeros, NA. We essentially assume that below this cutoff, the intensities are not reliable and we should do something else about them than just using them naively. And this cutoff is very simple. We look at the median, we look at the 97th quantile deviation from the median to the right, we flip this distance to the left. So it's very simple heuristic. But we're saying that this is the cutoff for the data set below which we do not trust the values. And so now this is how we think about this kind of from the statistical perspective. This is one protein in this data set. We think about this in terms of this data structure. Columns here are mass spectrometry runs. Again, remember it's label free. And so each, like several runs can be grouped because they're technical replicates on the same subject. The subjects can be grouped because they're biological replicates of a condition, and then we can have multiple conditions. Alternatively, we can have a time course or repeated measurements experiment. Now then this subject from condition 1 will be the same as the subject from condition 2. This is where MSstats requires a convention where each biological replicate has a unique identifier. If you have subject 1 profiled across multiple conditions or multiple time points, MSstats will see that and will recognize that we have repeated measurements or time course design. On the other hand, if each condition has distinct subject IDs, which are not the same, then we will recognize that this is a group comparison design and we do not have repeated measurements. And MSstats will fit the right model for that. So the pink part of this data structure is essentially biology, right? And the experimental design that we have. Now, the yellow part is the technology. In each run, we acquire multiple features. And this Y here is the log intensity of this feature. And then for some features, we have missing values and some features are below this cutoff. In statistical language, it's called sensory. And so now our goal is to summarize this data structure and do statistical testing in a way that where we build the signal to noise ratio, but in a more smart way where we understand what the nature of replication is. We represent peak intensities and read between the lines and say these are the values which should be censored. And also, well, some of these features have outliers and so on, and we need to be robust to that. If we have statisticians in the audience, just as a parenthesis, this is how traditionally we think about this type of data structures. So this is called a split plot design, where randomization occurs. We randomize the order of the runs at the columns, so in the pink part of this data structure. And then the features that we have, they affect the intensities that we observe, but they're not randomized. All the features are acquired simultaneously within the run. Because there's a restriction on randomization, we have two parts of this experimental design that we need to handle separately. And in the classical statistical literature, there is this type of general model, which is called a split plot model, which we can write. And if we had no missing values, no outliers, no every subject has the same number of replicates and so on, then it can be shown that conclusions regarding the biology, so comparisons between conditions, only depend on some kind of summary of all the features in the run. So we only need to understand the run level values to do the statistical inference, which gave us a thought that a two-step procedure which first summarizes all the features in the run and then focuses on the pink part of this data structures is more effective. And this is not a novel idea. Many people summarize features in the run and then do hypotheses, but being able to put it in the context of a more general statistical literature helps us make extensions which are much more robust and handle extreme cases in much more reliably. So specifically what we do, we first focus on this yellow part of the data structure, and we say, okay, if some of these things were censored, we have this indicator saying that we have a missing value, but the missing value is informative, meaning it's small. If it is missing, but it's not randomly missing, it is missing for reasons of low abundance. And then it allows us to summarize this type of data structure in a manner which is very classical kind of statistical way that handles censored data. If you are familiar with this, you can recognize this. If not, all we need to do to say is that we know that the features are missing for reasons of low abundance. What I can do though is I can give you the intuition of what's going on with this type of summary. So let's say we have two mass spectrometry runs. Let's say this is healthy and this is disease. And let's say that this protein has 3 features. This is what would happen if we could quantify all 3 features in every run. So this will be the average of the healthy, this will be the average of the disease. We say, okay, that's how the protein is regulated. Now let's say that this is the limit of quantification, and this particular feature cannot be quantified if we have, if the protein is at low abundance. So we have some choices. So for example, we can leave it missing. So then we will summarize what we observe in the healthy and then summarize what we observe in the disease. We see how we would underestimate the effect of regulation because we tend to have higher values observed and ignore that a lower value was missing. So we underestimate the effect of regulation, and we can even not be able to say that this protein is differentially abundant. What we can also do, we can impute with 0 over some other small value or integrate the noise in the data. Then we will overestimate the extent of regulation, right? Because now one of the values is smaller than it should have been. And so now this can potentially lead to false positives. So what MSstats is doing, and this is what this kind of mathematics really imply, is that we assume that the features supposed to be parallel because they come from the same protein. We expect all these features to be following the same pattern. And so for this missing value, if we have other features present in this run, and if we have this particular feature present in some other runs, then we essentially propagate this kind of by parallel pattern. We say this value is imputed, and so now we estimate the missing value more accurately. So this is what this imputation actually is. And then we will summarize this, and instead of taking the mean, we use a robust summarization to account for outliers. And so now after we have done this summarization, we have this simplified data structure where for each run, we have one number per protein. And so now that we have one number per protein, we can go back to the biological aspects of experimental design and focus on what is the type of replicates, what is the type of conditions, how can we build essentially the signal to noise ratio to have the most accurate p-values. So that's what MSstats is doing. And there have been a lot of benchmarking. Mina in particular has done a lot of work benchmarking this against various methods. This particular figure illustrates how this imputation plus robust summarization by Twiki median polish allows us to have accurate estimation. So this is one of the control mixtures where we know the true fold change. This is a particular protein, so the true fold change is 2. So the true log fold change is 1. And so this is what happens with the estimation if we have, and for 2 data processing tools. And so this is what happens if we ignore imputation or if we ignore robust summarization. This is what happens if we just take the sum of the features without doing kind of the statistically kind of complicated, more sophisticated things that we do. And so fundamentally, the number of comparisons and the estimations are more accurately if we do that. Now, to preempt questions, so we have noticed something more recently, that this type of assumptions is not always the right thing to do. For example, if you have post-translational modifications, and this is the work by Devon more recently, then we may not have enough data to do this type of imputation. It may also be possible that in PTM, the missing values are not necessarily due to low abundance. They may be due just to the enrichment step not working. So the protein, the PTM is there, but it just was not captured by the enrichment step. So there are clearly situations when assuming that missing values are due to reasons of low abundance is not appropriate. As I was saying earlier, max quant is very generous with missing values, so those missing values may come from other situations. Well, we always have options just to leave missing NA, in which case we will have something like this. I would say more work is needed, especially for these more recent workflows and PTMs and so on. So we will be certainly working on that. For now, this type of imputation is an option. So you can use it or you can leave it out if you would like. But in terms of benchmarking, let me get back to that. So this is 3 data sets, which are all spike-in mixtures where we know the ground truth. And this is, for each data set, we process this either with MSstats or just taking the sum of all the intensities of the features and then taking the log. And so what we're interested in is in reproducibility of the conclusions between the tools. Remember in the beginning, I said, we want to make sure that our conclusions reflect the sample composition, reflect the biology, and not the technology. And so what is good to see is that if we use this type of analysis with MSstats, the intersection of true positives between the tools is higher than the intersection if we just take the sum of the features and take the logs. And the same in terms of the false positives, we minimize the false positives compared to log sum. So these are 3 data sets, but we've done quite a bit more, and we're yet to see a data set where this is not the case. So the conclusion from this part is that if we interpret the data kind of more smartly and read between the lines, our conclusions become less dependent on the specific choice of the data processing tool that we use. So this is the first part, and now the remaining ones, I just take 15 minutes to just walk you briefly through some other functionalities that we have. So the differences in data processing. So this was also very useful for us to see. So this is the case study with a DIA case study, which you will actually work with in the hands-on right after the break. And you already worked to some extent with Brandon last week. Let me just review a little bit what was done for this particular experiment. So this was yeast samples exposed to osmotic stress, so the stress where yeast is progressively starved. There were 6 time points, 3 biological replicates per time point, so 18 DIA runs. And there were 2 analyses done by Skyline. The first one, just kind of traditional analysis, where for each precursor, we will select top 6 fragments and we keep everything. And then what is interesting in this data set, and that's why we like to use it for teaching also, is that the authors also did something else. They had a separate experiment where they had 8 technical DIA runs of a same sample to essentially understand how reproducibly and consistently they can quantify the same thing. And so from these technical refinement runs, what they did, they selected peptide precursors which were detected consistently. So detected in more than 50% of the runs, and also had low CV, so CV less than 20%. And so now we have a choice. Either we quantify everything we see, or we rely on this type of additional filtering based on prior experiment to only keep quote unquote good quality data. And so the question is, does it matter? Should we do this refinement runs, or should we somehow otherwise make sure that our data are good quality? And then after that, so we processed with Skyline, regular low CV. The data were also processed with Spectronaut and back then with DIA Empire. After that, we used consistent downstream statistical analysis with MSstats, but having this additional Q filter, so quality of the identification cutoff. So this is a comparison between the data sets. So this is when we work with all the precursors, and this is where we only work with precursors which have low CV. Well, it's a real life experiment. We do not know what the truth is. We do not know what to expect, but a couple points to note. So low CV clearly lost some proteins. We have 400 proteins fewer. But we can also see that for all the data sets, the patterns of changes in time across, as compared to time zero, is kind of jumpy, right? So it goes from 74 proteins to 300, then to 519, then down to 500, then up to 700. Whereas with low CV, things are much more consistently progressing. 58, 260, 373, 465, 700. I would argue that biology is more likely to be kind of smooth and consistent rather than jumping up and down. And so even though we lost some proteins, I'm guessing that this is probably more reliable than this type of differential abundance. Now, if we focus on a particular time point, we compare time 60 versus 0. Well, quite a bit of agreement between them, but also some proteins were detected as changing only with all precursors and some only with low CV. So it matters, right? The data processing matters. And this is one particular protein. And so we see what happens, right? So if we have low CV data, well, there's an outlier, but MSstats can handle this outlier easily, right, with this data processing. Whereas here, well, there's a lot more outliers, which will be somehow, eliminated or imputed. So it's a more difficult situation. And so we see that because of that, the conclusions actually change, right? So in this particular case, using all precursors, the protein is borderline significant. But then if you use good quality data, then you say, yeah, maybe not such a strong evidence for change. So what's the right answer, right? We do not know. It's hard to tell. But this definitely matters. But now, thinking about comparing for the same data set across different tools, we also see the same thing, that the agreement between the tools. So this is one particular protein quantified by 3 different tools. DIA Empire, we probably should not give much weight to that because FragPipe is much better now. Back then it was not designed for this type of instruments, right? But we see that the nature of quantification between Skyline and Spectronaut is somewhat different. And we see that regardless, the agreement between the tools is much higher if we do the processing with MSstats as compared to the processing with taking the sum and then taking the log. So again, by handling outliers, the difference between the tools is smaller. So this is one vignette. So we can kind of work with the data processing matters. Some extent of outliers we can handle, in which case we have less differences between the tools. However, there's no magic, and some of the upstream data processing definitely has impact on our conclusions. So this observation, particularly with this data set, motivated another project in the lab where we asked ourselves, do we have to have this low refinement runs to understand what it means to have quality data? Or can we somehow learn what it means to have quality data computationally without having to do additional experimentation? And so this is now the next step for that. And so this was a separate work. So the data is still by Brendan and Min. I think I have the acknowledgement. Yes, this is the paper by Tsungheng Tsai, which was published a couple of years ago in MCP. So that's what was the thought process. So let's kind of continue with this low CV data set and try to add another option of data processing. So we had 4 data sets which were resulted from different processing of the original data. So full sparse means we consider all precursors, use 6 fragments per precursor, and we only eliminate individual peaks if they do not pass the Q value quality of identification threshold. Or we can have full data set, again, all precursors, 6 fragments, and if half of the peaks in the feature do not pass the Q value cutoff, eliminate the entire feature. Right, so now here essentially we have more features, more missing values. Here we have fewer features and just remove the entire feature if it has too many missing values. And the same thing for low CV. Now let's just take this quote unquote good quality precursors and just introduce some missing values if there is a problem with Q value cutoff. And then let's eliminate the entire features if they do not pass the Q value cutoff. And well, it does matter. So in terms of the number of proteins, so clearly low CV and removing entire features makes us lose more proteins compared to full sparse. But full sparse will have more missing values and more noise, right? So somewhere there, there is some kind of middle ground where we need to keep just the right amount of good quality data and not eliminate too much. And so the question was, how can we computationally find what's this middle ground and hopefully make our conclusions less dependent on this type of subjective choices. So this is what the data look like. So this was full sparse. You see that those empty circles, they indicate missing values, and we have a lot of outliers because the fragments are not quantified reliably. Here is when we remove some of the features with too many missing values, you see that it becomes cleaner. And this is the low CV sparse. So now more outliers are eliminated just because we know that these are good quality data from prior experimentation. And we also see that, well, it certainly matters in terms of detecting differentially abundant proteins. So for example, comparing the first time point, we see that it varies from 58 to 147 differentially abundant proteins, right? And here it also varies, let's say time point number 4 from 500 to 800, right? So that's also quite a big difference. And this is the same processing with MSstats, so same statistical analysis. So these are really the differences due to the upstream data processing. And so the question for us was, how can we make sure that this happens as little as possible? How can we computationally clean this up so that we rely on good quality data without requiring additional experiments? So this was the work by Tsung Heng. He had a pipeline, which is now part of the MSstats software implementation. So the first thing is he detects low coverage features. So here we said, okay, 50% missing means low coverage, we remove. But why 50%? Maybe it's 40%, maybe it's 60%. So Tsung Heng was looking at the distributions of missing values in the data set and using kind of more objective statistical arguments to say this is to make more features, more missing values in these features than we would expect on average in this experiment. So these features have low coverage, whereas these other features have missing values, but it's consistent with the rest of the data. We should not remove it. Then what we need to do is to estimate the protein profile on average. So what do we expect in some robust way? What do we expect for this protein in general from the available data? And from this profile, we can find what are the individual outlying values. Now that we, and also again using some statistical argument, now that we detected missing values and outliers, there is an additional argument to say these features are overall too noisy, right? They have outliers, they have larger noise than what we expect. So we essentially look at the distributions of variances of the individual features. And this allows us to eliminate features with low coverage, detect outliers, and detect features which are overall too noisy computationally. Now, let me also kind of say right away, so we tried this, and because MSstats works with different acquisitions, we had to try it with different acquisitions. We tried this with DIA, DDA, and SRM. This type of workflow works best with DIA because we have a lot of features to learn protein profiles, to learn this, how much variation we expect to see, and so on. With DDA, it works to some extent if we have enough features in the protein. For SRM or like targeted experiments with few features, the main benefit really is kind of like finding individual peaks, which are outliers or data curation. It complements manual data curation, but I would say it's most useful for DIA. And so here is the example. So this is for a individual protein. Again, that's what we looked at. Now, this is how our conclusions become more robust to these choices of filtering, right? So this is if we use all the features. So we see that for a comparing time 2 versus time 0, depending how you process the data, there will be different proteins claimed as differentially abundant. And we see that now with this extra cleanup, the intersection between different methods is much larger. Meaning with this cleanup, it doesn't matter as much how you choose these processing parameters, you will have a high agreement. It's still not the same, data processing still matters, but at least we have less dependency on the subjective choices of data processing upstream. And the same question as before, so how does it depend on the choice of data processing tools, not just like the filtering parameters, but the actual data processing tools? Here is, again, for one of the data sets processed with 4 different tools. Gray is the number of features per protein before filtering. Orange is how many features we kept after filtering. So for all the tools, the number of features was reduced to some extent. And so now this is the agreement between the tools. If we use all the features, this is what happens if we use top 3 features, it's less agreement. This is if what happens if we have used top 10 features, more or less the same, but the agreement is higher if we use this type of intelligent filtering and data-driven filtering. So again, this type of data cleanup make us less dependent on the choice of the data processing tool. And so the last thing, it's kind of very brief, just to talk about the utility of these converters. So you can see already that because we have converters from multiple tools, it is easy for us to use standardized analysis and try different analysis strategies on different outputs and understand what can be done statistically versus what should be improved at the tool level. By the way, I need to mention that Mina has been interacting with all the developers of all these tools, and in the process, I think we debugged a lot of these tools as well, because we would find some mistakes, or they would be interested in knowing why certain proteins behaved certain way for their particular tool. And so because of these converters, we can compare not just the tools, but we can also compare various statistical methods, and we start doing this. We haven't quite finished that yet. But there, of course, MSstats is not the only tool out there. There are many other tools. So some tools that we found in the literature which do certain things that MSstats does. I would say that MSstats is the most general of all in terms of the inputs that we take, compatibility with data processing tools, but also in terms of experimental designs that we can handle. And for that reason, by the way, MSstats is more or less the default strategy now in both PRIDE and Massive because of the versatility of the analysis. And let me actually advertise right away. The day after tomorrow, we'll have a discussion of all these repositories and how we can work with historical quantitative data. But for now, I can just say that it allows us to compare MSstats strategy also with strategies by other statistical analysis methods in terms of various techniques. So here we look at the positive predictive value. We can look in terms of the true positives and false positives. We can look into this in details if you would like, but long story short, it's not uniformly superior, but more generally, MSstats is the most versatile and the most accurate tool of all across the board. There are some individual circumstances when it's not the case, but generally speaking, it does very well. And so the same thing in terms of false positives and false negatives, we can say, okay, how across different tools, how is the intersection. So we don't necessarily have the larger intersection as compared to the other statistical methods, but it is typically compensated by the smaller false positives. So not necessarily the most sensitive, but more sensitive tools will also have more false positives. So that when we develop our methods, we need to balance the sensitivity with false positive rate. So I'm happy to talk more about this, either answering Q&A in the chat or just offline after that as well. So this is pretty much the end of our hour. So let me just finish by acknowledging a lot of people who contributed to all of this project. So I again would like to highlight Mina, who was really the leader of all of this effort for a very long time. Ting, who has done work on the TMT-based labeling. We'll talk about this tomorrow. Cyril has worked on assay characterization. Manaza and Devon have done a lot of work. Devon for PTM and Manaza and Devon together on the Shiny interface. Tsung Heng, who did a lot of work on this feature selection work. And of course, Mateusz, who is currently the lead developer. So a lot of the efficiencies in data processing and in modeling are all due to Mateusz's effort. And of course, I would like to thank the collaborators from ETH, from University of Washington, from Roche, and from Genentech, because their feedback, and from UCSD, their feedback really has driven a lot of our work. So thank you very much. I am happy to answer questions if there is anything that I should answer maybe live with everyone. If not, I will stay online and I will answer some questions just by typing.